Enzymes are responsible for all the biochemical reactions that take place in our body. Enzymes are defined as a globular protein that increases the rate of biochemical reactions without themselves being used up. In other words, enzymes are biological molecules that change a substrate into a product and remain unchanged. Enzymes are very specific in their ability to convert reactants into products. However, a single enzyme is responsible for a single type of reaction that takes place in our body. Enzymes increase the efficiency of a reaction and do not affect the end product of the reaction. If a reaction takes place by an enzyme and the other does not, the rate of reaction in which enzymes participate is converted into a product first by using less energy compared to a reaction not taken by the enzyme. The minimum amount of energy required to start a reaction is called the activation energy of enzymes. The active site is a specific part of an enzyme where the substrate binds to start a chemical reaction in the body. The active site is made up of two definite regions. The first is the binding site and the second is the catalytic site. The binding site is the region of the active site where amino acids make temporary bonds with the substrate. On the other hand, some amino acids are responsible for catalytic activity, therefore it is called catalytic site. Enzymes are biocatalysts produced in the protoplasm and synthesized in the cell. They are much greater in size than the substrate. Besides the active site, some enzymes are completely made up of protein and non-protein parts. These enzymes are called conjugated enzymes. The non-protein part of the enzyme, called the cofactor, is responsible for the attachment of the substrate at the active site and performs catalytic activity. The conjugated enzyme without a cofactor is called an apoenzyme. It does not work separately. The lock and key model depicts enzyme-substrate interaction as a static, preformed active site fitting the substrate precisely, like a key fitting into a lock. It proposes that enzymes and substrates have specific shapes, ensuring accurate binding without significant conformational changes. This model simplifies enzyme-substrate specificity, suggesting that the enzyme's active site remains unchanged upon substrate binding, facilitating efficient catalysis. The induced fit model proposes that enzymes undergo conformational changes upon substrate binding. Unlike the lock and key model, it suggests that the enzyme's active site is flexible and adjusts its shape to accommodate the substrate. This dynamic interaction enhances the complementary fit between enzyme and substrate, optimizing catalytic efficiency. The induced fit model highlights the dynamic nature of enzyme-substrate interactions, emphasizing the mutual adaptation of both enzyme and substrate during catalysis for efficient chemical transformation. For following factors can inhibit enzymes: 1. Substrate concentration. 2. Enzyme concentration. 3. pH. And 4. Temperature. The amount of enzyme present in a reaction is measured by the activity it catalyzes. The relationship between activity and concentration is affected by many factors, including temperature, pH, and substrate concentration. The concentration of the enzyme is important in chemical reactions as it is needed to react with the substrate. Often, a small amount of enzyme can consume a large amount of substrate. Substrate concentration refers to the amount of substrate present that can be converted into a product. It is typically measured in molarity, moles per liter. Increasing substrate concentration generally leads to an increase in the rate of enzyme-catalyzed reactions. This is because a higher concentration of substrate increases the likelihood of collisions between enzyme and substrate molecules. The pH scale is used to measure the acidity or alkalinity of a sample, affecting enzymatic reactions. Enzymes have an optimum pH where they are most active. pH changes can lead to denaturation or slow down enzyme activity. Enzymes are proteins with a specific three-dimensional structure influenced by the charges of amino acids. pH affects enzyme activity, with each enzyme having an optimum pH range. Extreme pH values can cause denaturation. Most enzymes have an optimum pH between 6 and 8, where they exhibit high activity. Deviation from this range may result in reduced enzyme function. Temperature has a significant impact on enzyme activity. As temperature increases, enzyme activity generally increases until an optimal temperature is reached. Beyond this optimal temperature, enzyme activity decreases as the enzyme becomes denatured. At low temperatures, enzyme activity is reduced. This is because the enzyme molecules have less kinetic energy and are less likely to collide with substrate molecules. At high temperatures, enzyme activity decreases. This is because the enzyme molecules become denatured, which means that their shape changes and they can no longer function properly. Enzymes are typically classified based on their function, structure, and the type of reaction they catalyze. The following are the classifications of enzymes. Oxidoreductases. These enzymes catalyze oxidation-reduction reactions, involving the transfer of electrons between molecules. B. Transferases. Transferases facilitate the transfer of functional groups, such as methyl, acyl, or phosphate groups, between molecules. C. Hydrolases. Hydrolases catalyze hydrolysis reactions, where a molecule is cleaved by the addition of water. D. Lyases. Lyases catalyze the removal of groups from substrates by mechanisms other than hydrolysis or oxidation, often resulting in the formation of double bonds. E. Isomerases. Isomerases catalyze the rearrangement of atoms within a molecule, leading to the formation of isomeric forms. F. Ligases. Ligases, also known as synthetases, catalyze the joining of two molecules, often coupled with the hydrolysis of ATP. I hope you like the video. Thanks for watching.